Okay, we just described all the organizational levels of the skeletal muscle, from the muscle fascicle, muscle fiber, then myofibrils containing the myofilaments in the previous video. And we have two types of myofilaments. The thin, these blue ones are the actin filaments, and the thick ones, the red ones, are myosin filaments. The structure and the detail of that is part of a physiology class. But in here, let's just keep in mind that the thin filaments bind calcium ions. This is important to understand the next part. And look how they neatly organized to form this structure that repeats over and over and over throughout the entire length of the skeletal muscle fiber. And each of these units is called a sarcomere. This is the functional unit of the skeletal muscle tissue. This is where contraction happens. I'm gonna draw this step by step so we can identify all the important aspects of the sarcomere. Let's start by, where should we start? Okay, let's start by, where is it? Okay, drawing a Z line, which marks the beginning, but also the end of a sarcomere. So this is the Z line or Z disc. Now, right in the midline of the sarcomere, remember, from one Z line to another Z line or Z disc, that's one sarcomere. So right in the middle of that sarcomere, we have a line. And since that line is located right in the middle, what if we call it the M line? M line. Easy. Now I'm going to use the same colors that your book uses to avoid any confusion in here. Let's start drawing the thin filaments. Thin filaments. Okay. Thin filaments start at the Z line and they go all the way towards the M line, but they stop there. Z line and they stop about here. Z line, they start here and they stop, and the last one, they stop in here. Same thing is happening on the other side. And just for the sake of an explanation in the next part, I'm gonna go over past the sarcomere. So I'm crossing the Z line, but please understand that one sarcomere begins and ends at a Z line. Okay, and in the other side, this one and this one. Now, this in blue are the actin. Myofilament, the thin ones, the ones that bind the calcium. Now, in red, we're gonna draw a thick filament. And this thick filament begins now in the M line and they move towards the Z line, but they stop. They don't touch the Z disc. Same thing in here. So they're touching the M line, but they're not touching the Z disc. And as you can see, they kind of overlap a little, the thin and thick filaments. And we want that. Now, these thick red filaments are myosin filaments or myofilaments. So easy so far. Now, this area in between or involving the thick filaments is called a band. A band. And see that in the A band we have, well, the M line, but we have also this area between the M line uh, containing on either side of the M line containing only the thick filaments, the myosin filaments. That is called the H line. This and this, H line. And this area between or containing both thick and thin filaments is called the area or the zone of overlapping. Okay, so A band, it has three components. Right in the middle, we have the M line. On either side of the M line, we have the H zone that contains only thick filaments, myosin filaments. And on either side of the H zone, we have the zone of overlap, where we have overlapping the thin and the thick filaments, actin and myosin filaments. Now, let's explain now the I band. And the I band consists of this area containing only the thin filaments. This area is the I band. And as you can see, the I band consists or contains thin or thick actin filaments from one sarcomere, but also actin filaments from the sarcomere next door. So this is the I band. And this pattern repeats, as you can see in here. This is another I band, this area. Okay, this pattern repeats and repeats and repeats. I band, A band, I band. This is what creates the striated pattern that we described later, that we see on the surface of the skeletal muscle fiber on the cytoplasm, sarcoplasm. So the I band, since it consists of thin filaments, it's lighter. When we see it, then the A band is darker because it's where the thick filaments are. Okay, now let's see that or these structures on a beautiful sarcomere, not drawn by me. And am I okay? So again, this is the Z. This is one myofibril. It's not a muscle fiber. It's a one myofibril containing thin and thick filaments. So in blue, we have the thin filaments, actin, and in red, we have the thick filaments or myosin. In here, the blue and the red lines. Now, one sarcomere is just in between one Z disc and another Z disc. So this from here to here is one sarcomere. Right in the midline, we have the M line. Look at the myosin filaments, how they touch both on either side the M line, but they don't reach the Z lines. Now, there is one very important, huge protein in here that is called the titin. And the titin is a filament that attaches to the Z line, runs through the myosin filament towards the M line on both sides. So it attaches the thick myosin filaments to the sarcomere and gives stability to this structure. One important thing, and you can see there is like a coiled protein. It looks like a spring. When the muscle stretches, this protein uncoils or stretches too. And when it contracts or the muscle goes back to the original position, it recoils again the titin protein. So this provides elasticity to the muscle. Now, one important function is that it resists the overstretching of the muscle to a dangerous point where we can just uh, rupture or injure the muscle. How? Because the as it uncoils when we are stretching the skeletal muscle fiber and the entire skeletal muscle, it becomes stiffer and stronger, trying to resist that overstretching placed on the muscle. What else can we see in here? Well, the rest of the elements, the thin or actin filaments from the Z line, but they stop before reaching the M line on either side. So in that way, we create the two different bands, the A band containing thick filaments and thin filaments. Remember that we have the three components, the M line on the middle, the H zone on either side is this area containing only the thick filaments, and lateral to that, the zone of overlap, where the thin and the thick filaments are close together and overlapping. That's the A band. On either side of the A band, we're going to find an I band, which includes two different sarcomeres. Actin filaments from one sarcomere, actin filaments from another sarcomere, and right in the middle, a Z disc. Important to have very clear this concept as we explain in the next video how the sarcomere contracts and how this element changes in a relaxed and in a contracted sarcomere. Now, before finishing this video, I want to show you, I want to show you how this sarcomere, or the relation of this sarcomere with the other organelles inside the muscle fiber. So let's get oriented in here. This is a muscle fiber. See, we're peeling here the sarcolemma. And remember that we can poke, look in this image in here, we can poke the sarcolemma in towards the sarcoplasm, creating these invaginations, the T-tubules, the yellow ones. See, and see the openings in here. This is important because the action potential, the electrical impulse coming from the neuron on the nervous system activates or stimulates the cell membrane, the sarcolemma of this muscle fiber. And the only way we can ensure that the entire, all the myofibrils are going to be stimulated to contract, and not only the ones closer to the cell membrane, is by poking or creating these T-tubules that reach deeper myofibrils inside the sarcoplasm. Now, again, inside the sarcoplasm, remember, of the muscle fiber, we have a bunch of myofibrils, and each myofibril consists of thin and thick filaments, and they are arranged like this, forming the sarcomere. Now, the place where the triad, that structure that we described before, consisting on a T-tubule and a terminal cisternon on either side, that structure, the triad, is placed right on top of the overlapping, in between the zone of overlap, in between the A and the I band, right here. Why? Because remember that the terminal cisternae is the one containing the calcium ions, and the T-tubule is the one is going to be stimulated, or where the action potential is going to travel. So we are right there. The calcium ions can be released right there into the zone of overlap, where we have the thin and the thick filaments to interact and produce muscle contraction. So see you in the next video to explain a little bit more of this process.